Luego, bueno, hizo la tesis conmigo y ahora ya está hace un tiempo trabajando bueno, haciendo su posgrado en el Bernstein Center for Computational Neuroscience de Berlín. Es un centro muy reconocido a nivel mundial de neurociencia computacional, donde se dedica mucho a hacer modelos, redes y demás. Hay muchos de los grandes reconocidos mundialmente en estos temas. Y pues nada, hoy nos va a contar un poco de cosas que estuvo también trabajando conmigo y de redes también que ayudó a desarrollar en parte también en las cosas que fue aprendiendo en el último tiempo. Así que, en fin, bueno, muchas gracias Bernie por acompañarnos hoy y darnos la charla de tu trabajo. ¿Okay? Line, no sé si hay personas en inglés, hay que checar. ¿Eso, Bernie, hay gente en inglés? Sí, o como prefieras, si quieres darla en inglés tampoco es que produce demasiada. ¿La prefieres dar en inglés? Ok, bueno, buenísimo, gracias.
Okay. Okay, so good morning to all. My name is Bernardo Andrade. Ortega, as Professor Rossi already told us. And I'm going to be presenting my work, my bachelor's thesis that was named Bimodal Multiestability during Perceptual Detection in Premotor Cortex. So, see. Yeah, I'll try to speak a bit louder then. So first, I would like to start with an example. Let us imagine that we are this little mouse eating some cheese, minding our own business, but we have to be aware always of the environment. In this case, we have a predator lurking. So, as you may see, if we only use the sense of sight, it's fairly difficult to tell where the predator is. However, we can also use the information from other sensory modalities to aid us in this. For example, if we hear the predator approaching us, maybe it steps on the branch, we can use that information, or maybe we can perceive its smell. So the question that arises is, how does the brain manage to integrate this multimodal information when all of the sensory channels are competing within each other? And of course, how do we take this information then to make a decision? In this example will be whether to stay there eating the cheese or to run away. And just so you don't die without knowing, the predator here is a little cat, and even if I mark it with a red circle, it's really difficult to see. Okay then, so we won't be studying all of the brain at the same time. We will be focusing on one particular area, and we're interested in studying the ventral premotor cortex. Why? Well, in previous studies where they use tasks, where they only presented one modality, it was shown that the ventral premotor cortex shows tactile coding and acoustic coding. Plus, as many other premotor and frontal areas, the neurons within this area show working memory, and sensory stimulus processing. And to study this multimodal integration, we'll be using this bimodal detection task that was previously studied, but yeah.
Well, something that is important to say here is that this cortex is receive a lot of input from parietal cortex, like somatosensory, visual cortex. So if you look like the connectivity graph or the connectivity matrix between BPC and other areas, this apparently is an area that receives so many input from a lot of sensory areas and a lot of premotor cortex too. So its communicate is like in a hub position to integrate, to give this perceptual or modal coding. And something that is important to remark that in human, this area became some area that is so important for language. So it's the Broca area that we employed a lot, and we have a lot of connectivity between a lot of sensory inputs to create the language and to communicate. So it's apparently a key area. I don't know how, if it's redundant with another area, I don't know, but at least all the data that we have show us that this is a really key area for integrating signals.
Yeah. Thanks for the addition there. Okay, so continuing, we employed this bimodal detection task that was previously studied by one of my colleagues in the lab, Dr. Parra. And the task basically consists of this. We have our subject, in this case, a macaque, that's in a setup similar to this one. And it's holding a lever with his left hand and being stimulated in the right hand. And the task timing is this one over here. So the subject is ready to continue the trial during this point, and we have a pre-stimulus time that is of a different duration in every trial. And later we have the stimulation that can be tactile or acoustic or no stimulus at all. Later, after that delay period, the subject has to report which modality did the stimulus come from, or in the case that we didn't present anything, if it was absent, when pushing one of these three buttons. And the behavioral response can be classified as this. Here in green, we have the trials where the subject gives the correct response, the hits, and the correct rejections. But the subject can also miss our tactile or acoustic stimuli, or give a false alarm. That is when we don't present nothing, that the subject tells us that it failed tactile stimuli. And we also have this very interesting kind of error where we present a tactile stimuli and the subject reports an acoustic one. But during this study, we'll be focusing only in the correct trials, and we leave the incorrect trials for the studies.
What is MU?
MU is multimodal response, right? Something like that.
Okay, but I get the idea. Yes.
Yeah.
Yeah, it's like a misunderstanding, I think.
Yeah, misunderstanding. I think that's the coding there. Yeah.
So I have a question that even if I've been working with Sergio and with this, I don't have an idea what happened. Why do you believe that the animal understand so receiving tactile input, but he believed that it was an acoustic or the other way around? I don't know. Why do you believe that?
Well, this is very hard to interpret, but maybe the subject was not paying a lot of attention during the trial, So he received a stimulus, but then he kind of forgot or didn't care about which one. But he remembered that he perceived a stimulus, so he just randomly presses the button here. Yeah. Yep. Okay, I'm not sure how to see questions here. Okay, over here, yeah. And miss is the same. No, well, miss is when we. Oh yeah, of course. So the question says, so mu and miss is the same. Am I right? Yeah, and the answer is no, because the miss is when we don't present anything, and sorry, when we present a stimulus, tactile or acoustic, and the subject reports that it didn't feel anything. MU is when we present, for example, a tactile stimuli, a stimulus, and the subject reports he perceived an acoustic stimulus. So he confused the modalities. Yeah.
Yeah, no, I do really, well, probably the reason, I'm not so sure, the reason why this misunderstanding trial appear probably similar to the false alarm trials. I'm not sure. That false alarm is when you don't receive nothing, but you imagine that you receive something acoustic or tactile. So in that sense, in some ways, quite similar, because you receive something that is tactile but is quite low in amplitude, and you believe that it was tactile. We need to check if, or we should check if the accused or of the misunderstanding are.
Yeah.
Yeah, that I understand what you said is that in the MU, they are confused between the two modalities, right?
Yeah.
And in the other one, it's just, there is no this confusion.
Yeah, yeah, exactly. Yeah.
And is there also a possibility that it's just a mistake in which button, like he forgot which button means what, and so he's pressing the tactile button thinking that that's the acoustic one? Could be.
I find that very unlikely because
Yes?
the animal is like highly trained.
Okay.
So I think he has a good representation of what each button
Okay.
is. On chat, okay. So the questions reads, does the MU happen when with the same probability regardless of the difficulty of the stimulus? Or if it's like a low amplitude stimulus, for example, it's more likely to get confused. Yeah, these mistakes happen mainly with low amplitude stimuli. Yeah, so one thing that I didn't mention yet is that we vary the intensity of the tactile and acoustic stimuli across the trials, but they're very close to the decision boundary, the detection boundary, sorry. And yeah, we get all of these mistakes with very low amplitude stimuli. Yeah. So I think with this we can proceed to see the performance of the subjects between two monkeys. And in here we see the psychometric curves. In the x-axis we have the amplitude for the tactile and the acoustic stimuli, and in the y-axis we have the probability of detecting it. Basically, we're saying yes, I felt the stimulus. And we can see that these are very good sigmoidal shapes, and this is related to what we see in the unimodal detection task. For example, the task when we only present the tactile stimulus, it looks like this. And yeah, it's not precisely the same curve, but let's remember that we have different physical units here. So making like a one-to-one comparison is hard. And yeah, so what my colleague found mainly is that in sensory areas, that is in area 3B and in area 1, the neurons respond only to one modality. Here, the tactile modality. So here on the y-axis, we see the information carried by the neurons, and on the x-axis, the time. And as we can see, during the stimulation, this area is very fast to respond and only responds to tactile stimuli and does not carry any information at all about acoustic stimulus.
Well, in relation with the question that we were having before, I think that in the previous slide,
yep,
if you well, we weren't talking before is that this well, here we only see false alarm, but the zero amplitude that sometimes the animal responds yes. Well, these are false alarms. So
yeah, exactly.
Yeah. But on the other hand, the ones that are so low and the animal usually says no, well, are quite similar to a false alarm because it's something that is substressable. So it cannot be perceived for the animal. So probably, I'm not so sure because it hasn't been checked, but probably if someone analyzed the misunderstanding trials are in that range. I'm not so sure what happened there. I don't know.
Yeah, we have to check the responses in those trials. Okay. So continuing, we get this. So sensory areas only seem to process information from one modality. And this is true, well, at least in monkeys. In mice, I think there's been studies where they show that the visual cortex actually processes response to information of more than one modality. But as we're talking about monkeys here, we can say this fairly confidently. So as I showed you before, S1 responds mainly to tactile stimulus. And we can assume that something similar happens for the auditory area 1, A1, that responds only to acoustic stimulus. So we need to, to perform this task, we need an integrator. And because of the connectivity that Professor Rossi told us and the stuff I presented before, we think the ventral premotor cortex is a good candidate for this area that integrates information of both modalities. So without further ado, we can proceed to the individual neuron responses of the ventral premotor cortex. So first, I'll show you 3 neurons that we obtained. I plot here the raster. For anyone who doesn't know, raster plot, here we have in the x-axis the time, and in the y-axis the different trials, sorted by class. So here we have the absent trials, here the acoustic trials of the varying amplitudes, and here the tactile trials. Yeah, and over here, down, we have the firing rate of these neurons, which is basically the average for all the trials of one class. And as we can see, we have a very wide range of the responses in only these 3 neurons. So the first neuron over there on the left mainly responds to acoustic stimulus, while the second neuron, this one in the middle, responds more to tactile stimulus and only during the stimulation period. While this neuron over here on the right seems to show a more complex response. At first, it appears to respond more to acoustic stimulus negatively, that is decreasing the firing rate. But then it actually, the trajectories are actually separated for the null, the absent trials, the tactile and the acoustic trials. And then the acoustic and the tactile converge. And during this period, we could say that the neuron is basically coding for I felt something, regardless of if it was tactile or acoustic, or I didn't feel anything. And we also see some dynamics here after the probe up. For that, I have all the 3 examples. So these are all the 3 neurons. The one on the left shows a positive response for acoustic trials, while it shows a negative response for tactile trials. And it also shows a very clear ramping over here in the absent trials. As you can see, the firing rate does not remain constant during time. The next neuron, the one in the middle, responds firstly positively to only tactile stimulus, stimuli, and then negatively in the delay period. And this last one over here, yep.
What the colors are from? The blue one and the other one?
Yeah, this is the blue ones correspond to the to the tactile stimulus, and the orange to the acoustic ones. And these distinct shades of the color represent the distinct amplitudes. Yeah. So this last neuron over here on the right mainly shows responses after the probe up, and yeah, it increases the firing rate for only the acoustic trials. And like these 6 neurons, we have over 900 more neurons like this that we recorded and sorted. So the next thing we wanted to do is to quantify better these responses that I described to you right now, just qualitatively. And in order to do that, we have employed various metrics. First, I'll start with the variance. So the variance of the responses tells us basically how different are the responses for the different classes. Here we grouped the trials. First, the variance within the tactile trials, including the no stimulus or the 0 amplitude. The variance for the acoustic trials in a similar fashion to the tactile, and the decision variance, that is taking the mean of all the trials with a tactile stimulus, an acoustic stimulus, and no stimulus, and taking the variance between those 3 points for each time. And we can see that the neurons respond strongly to tactile, to the tactile stimulus, but also respond to the acoustic stimulus with a little delay. The tactile comes first, and then the acoustic, and the acoustic is in the same intensity. Then the variance of all of these 3 gets reduced during the delay period and increases again after the probe up. And we also obtain the temporal variance. This variance is obtained by taking a time window in the basal activity, for example, over here, and comparing the mean firing rate across all trials with this basal window. And we can see that there's an increase of variance just as the stimulus arrives and after the probe up. And here the variance rises significantly more after the probe up, but this could be because we are averaging also with the absent trials. So when we don't present anything at all, here we don't have any stimulus, but here we always have a stimulus, regardless of tactile, acoustic, or no stimulus. So therefore, the variance is more.
So Bernie, based on that dynamics that you are finding here, what's your opinion? What do you believe about the role that this BPC area is having on this task? Or what is the main role that you believe?
Yeah, with these first results, we can see that the BPC is like mainly a first sensory integrator. And with the decrease in variance here during the delay, we can maybe assume that it doesn't play like that big of a role during the decision maintenance of the task. Okay, so this was regarding variance, and we also employed another metric that tells us the information that's been carrying, that each neuron carries with its firing rate. And this is the mutual information. So basically, for each time beam, we computed this metric, and for each neuron. And if it was significant, we consider the neuron in this graph. So this is the percentage of neurons that carry a significant amount of information. This tells us that the neuron is separating at least one of the trajectories compared to the other ones. For example, the tactile to the acoustic and the non-stimulus one. And similar to what we saw in the variance, we see an increase after the stimulus that gets maintained and decreases a little bit during the delay, and later increases again.
Yep. What metric is this again? Can you?
Is the mutual information.
Mutual information. Okay.
Yeah. Over here, the pink trace represents the population on randomness. So basically, if we shuffle everything, how many neurons are expected to just code randomly? Yeah. And yeah, even though we see that the percentage of coding neurons decreases during the delay, it doesn't get near at all to the population randomness. Okay, so also employing this metric, we next wondered if we could classify the responses of the neurons. And instead of comparing the 3 different distributions all at the same time, we compare each pair of the distributions. So here we compare tactile with acoustic, no stimulus with tactile, and no stimulus with acoustic. And we further discretized this with a 1 or a 0 if it was significant or if it wasn't. Okay, so if the neuron separated all of these 3 possible responses, we classified it as a decision neuron. So, for example, I'll show you this neuron that we saw before. And for each time being, we employ this classification. So we can see here that it separates the no stimulus, the tactile, and the acoustic. So it's classified as a decision neuron. Likewise, if it separates the tactile from the acoustic one, the no stimulus from the tactile one, but it doesn't separate the no stimulus from the acoustic one, is classified as a tactile neuron. And similarly, we use this to classify it as an acoustic or as a no stimulus neuron. So for example, in this neuron here, we see, let's start with here, right? So the acoustic trials are separated, but not the tactile and the no stimulus one. So in this period, the neuron is an acoustic neuron. And in this period, the neuron is a no stimulus neuron. And a funny thing about these no stimulus neurons is that they can also be interpreted as stimulus presence or absence neurons, regardless of the modality. So as we saw before, here the neuron doesn't care if the stimulus was tactile or acoustic, it's only responding to the presence or absence of the stimulus. And if we see this for all the other neurons, then we get the amount of neurons that have this type of responses. We obtain this graph over here. So in the x-axis here we have the time and the percentage of neurons that are coding with each type of coding. So as we saw before, there's a great increase in the number of tactile neurons just after the stimulation, of the beginning of the stimulation, and later during the delay period, there's a wide range of types of coding, and it's actually kind of equivalent. So if we take this slide over here, and we plot, and not with the respect to the percentage of all the neurons, but only with the respect to the percentage of all the coding neurons, we get this over here. So out of all the coding neurons, around 60% are tactile, just after the end of the stimulus, and we also have the other types of coding, but less. But however, if we take this right in the middle of the delay here in the B section, we get this. And here we can see that it's kind of like proportional to all the distinct types of coding. It doesn't seem to show any preference for one. And we also get this blank space over here. That is all the neurons that we weren't able to classify with this. For example, if a neuron had a number 1, but here, but 0 and 0 here, we weren't able to determine the type of the neuron. And yeah, that corresponds to this white spot. Yep.
Just some questions to understand you. So in C, you have all the neurons, right? All your neurons?
Yeah.
Okay. And could you say again what a coding neuron is? How did you, just to follow you?
Yeah. A coding neuron is the one where we compare the three possible modalities decisions at a time, and if only one of this is enough separated from the other ones, then it's a coding neuron.
Now, the other question is, if you go back one slide, please, one slide back.
Yeah.
So I understand this is your geometric information measure in your
mutual information.
Sorry,
yeah.
Mutual information, that one. And so that's your y-axis, right?
Here?
Or what is your y-axis?
No, the y-axis is the firing rate. Yeah, so I kind of chopped off the axis.
So that's just only the firing rate?
Yeah, that's the firing rate.
So how do you use this mutual information for this? Just to get it?
Yeah, so basically for each time being here, we chop off like a slice, and we have a distribution of points for the tactile and the acoustic and then absent trails. And with this, we take distributions, and we take then we compare each pair of the distributions to see if they are different. And with that,
like
we
just separate them.
Yeah.
And, okay, to classify. But that's just okay, the fighting, right? Okay, thank you.
Yeah.
So yeah, important learning from here, Bernie, is that at least to have some equations, so for example, to show the people, because it's important that to explain these metrics. But the key is that you compare, as you were saying, the distribution between the two, the three conditions in this case, but you are pairing each other to one against the other one. So depending on the combination of which one is significant and which one is not, you can say, or you can classify, or you can label the different type of coding. So yeah, this is a little the idea of how he classified here the different codings. No, with a significant.
So there's a threshold for the mutual information, or how do you like assign the 1 or the 0?
Yeah, we employed permutations, and if it was significant, the information was greater than 95% of the time, I believe, than the permutation, then we assigned a 1. Question in the chat. Okay, Marcus says, I think there's still a preference in the B section. 34% is more than the double than 11%. Yeah. Yeah, so here, yeah, we can say that there's a slight preference for coding the stimulus presence or absence. Yeah.
I see that we haven't noticed before is that at the beginning there is more preference for coding tactile, and then at the middle of the decision delay there is more preference for absent coding. So yeah, it's quite interesting. Yeah.
Yeah. Okay. Oh yeah. One last thing I didn't explain of this figure is the G panel over here. So this is the response delay of the response onset of each neuron. So the first time each neuron was classified as one of these types, well, when did it happen? We have this to show. So yeah, it's basically how long does each coding takes to emerge. And we see here that the first coding to emerge is the tactile one, compared to the other ones that emerge just after the stimulus ends over here. And yeah, pretty similar for the rest, the session, acoustic, and no stimulus. But as we saw previously with variance, the tactile is the first one to emerge. Okay, so our conclusions for these individual neuron studies is that BPC neurons encode for both tactile and acoustic stimuli. And yeah, this is great. So our hypothesis was correct. The BPC responds to both modalities. And yeah, the next conclusion says that the BPC shows a faster and stronger response to tactile stimuli. We saw that with the variance and also with the mutual information response delay. And finally, the BPC neurons can show sensory responses, but also keep a decision memory. We saw that with the mutual information, right? That it didn't go down during the delay. Okay, so next we wanted to actually understand how does the network is able to process multimodal information and keep this decision memory. And for that, we employed population dynamics in the ventral premotor cortex. So the basic idea of the population dynamics is that we have a very high dimensional space n corresponding to the number of neurons. In this case, plus than 900. Yep.
Yes, sorry. I have two questions.
Yeah, go ahead.
The first one is, we see that it has like a big percentage of the neurons at the end of the stimuli are coding for tactile information, and then as times goes by, these codings changes, right? As you show in A and B.
Yeah.
Does this mean that some of the neurons that are coding tactile information are like transforming their coding properties as the task goes along?
Yeah, exactly. As we see with the example here, for example, this one, it starts as an acoustic neuron, but then it switches.
Okay.
So a similar thing happens for those neurons.
And the other question is, do you think that this like over-representation, I don't know if it's okay to call it an over-representation, but this big signal that we see of tactile information is something that is intrinsic to the BPC, or is something that has more to do with the experimental, let's say, gathering of the information of the neurons?
Yeah, both of those are good hypotheses, and I don't think we are able to say which one is the good one. Probably a bit of both. So the connectivity, the BPC is closer to the area S1, processing sensory tactile stimulus. So maybe that's influencing in this tactile bias, but also the way the task was trained. I believe they first trained the monkey to do only the tactile detection, and then they implement the acoustic detection as well. So maybe that's producing a bias in all of the networks to have a tactile preference.
Okay, thank you, Bernie.
Yep. Okay. So Marcus says, did you perform any kind of significance tests on the single neuron results? Yeah, so basically what we see here is the result of all the significant tests. So in panel D, every neuron that we show there is a neuron with significant mutual information, and same for panel C and F. So yeah, I think that answers the question. Yeah, the permutations test. Yeah.
Yes, so I think Bernie. Oh, okay. Something that is quite related with Beto's question is that, well, what you are seeing a lot of neurons that change the coding, and neurons that at the beginning are tactile, acoustic, and then they're like moving preference coding. What do you means for you? What's your explanation about that, or how? Yeah.
Why do they switch? Yeah. Yeah. I don't think we can like understand that in a single neuron level, and for that, like we can employ the populational dynamics.
Because neurons in sensory areas don't have this kind of flexibility. They just respond to one type of stimulus, right?
Yeah.
But these ones do, they can change what they're responding to. So that's the magic that's happening here, I think, right?
Yeah.
Okay.
And something that is related with all of that is that these neurons exhibit quite, quite crazy heterogeneity. It's like there are, we are seeing that here, but there are neurons doing a lot of things, something that is only tactile across the task, another one that are only acoustic, and another one that, they were changing, moving. So it's like a mix of different neurons that, still we don't understand why this is necessary for the network, but it's so hard from the only single neurons to understand, to interpret what this network is doing, something.
Okay, so moving along. This lets me, So we were on the population dynamics, and let's start from the top. So the basic idea of the population dynamics is that we have a very high dimensional space corresponding to the number of neurons, but we want to study it by reducing it to a low dimensional space. And this has been shown a lot in recent years, that although the neuron dimensional space is high dimensional, the activity lies in a low dimensional space that I usually love to call a manifold. And it's also useful for us because we can visualize it and study it in a better way. And for that, we used dimensionality reduction techniques. The main one, or the principal one in this area, is the principal component analysis, PCA. That's a linear projection related to variance, but we can also use other types of linear projections. Here in this study, we used the weights of a generalized linear model that I'll explain in further detail later. And we use also the mixed principal component analysis used by Kovac and coworkers. So starting with the PCA, we obtained, we applied the principal components analysis to all the tasks, and we obtained this. So these are the first 6 principal components. That is the components that explain the most variance. We have the projection over these components of all the population, basically the populational vector. And so starting from the top, in component number 1, we see mainly a tactile sensory response. In component number 2, we see
Bernie, I don't know if the people is familiar with that PCA. Probably we have here a lot of talk here about PCA, but how do you, in this case, how do you compute the covariance matrix that you employ for this calculation?
Okay, so to obtain PCA, we use the covariance matrix calculated with the covariance between each neuron, averaged across trials and time. Later, the eigenvectors of that matrix represent the principal components. Yeah, so following, the first component is mainly tactile, while the second one is mainly acoustic. But we also see like a tactile contamination in this component. It's not purely acoustic. Yeah, both of them are sensory, while the third, the fourth, and the fifth appear to be more of a decision maintenance. So here we see that during the delay, the stimulus presence and stimulus absence trials get separated in this third component. And here this fourth one is very static. We see a clear separation of the tactile, the no stimulus, and the acoustic responses. And here the fifth one is kind of similar to the third one, but with also a sensory component. And yeah, this sixth one again is kind of like acoustic, shows an acoustic sensory response, but also stuff going on during the delay. So what you can say here is, well, you just took an arbitrary number of components to show us, but how do we know if these components are significant? Yep. Yeah, so how do we know if these components are significant, or how do we know that we are only seeing half of the picture with these components? And for this, we actually calculated the significance of each of these components. So basically, to do this, we calculated a noise covariance matrix that's over here, the shuffle line, and we calculated it, I believe, 100 times. So we obtain this distribution. And if by principal components, the first one explains the most variance, and then as it goes on, it explains less and less variance. So we can see that at least these 6 principal components explain significant amount of variance compared to the just noisy data. And yeah, maybe we can also say that the 7-1 also explains some variance there, but yeah, we couldn't fit it here in a static fashion.
In the first component, do I also see a dependence on the strength of the stimulus? I think
some components.
The darker curves are like stronger stimulus. So those are coded like more strongly in the first component.
Yeah,
maybe in the fourth too.
Also as well. Yeah.
That's interesting. Yes.
Yeah, so that kind of relates to the sensory processing of the BPC and not just the decision of tactile stuff and acoustic stuff.
That's a great question. Thank you, Claudio, for that. Yeah, it's something that is important to remark that this area, at least at the beginning during the sensory part, it exhibit like a, I don't know, parametric coding or something like that is related with the strength of the amplitude, tactile or acoustic one. Yeah.
Okay, yeah. So what these results point us to is that there's kind of like a separation between the sensory dynamics and the decision maintenance dynamics. So we wanted to further study that. And for that, we employed the same analysis PCA, but only during a certain time frame of the task. So we separated the task into the stimulation period and the delay period. And we calculated the principal components here. Okay, so first, we see the projection here over the first 3 principal components during the stimulation period. And here we see the explained variance of these 3 components. And we see that the explained variance is mainly rise during the stimulus period, but also after the probe up, and remains low throughout the delay of these 3 components. And also, we see here that 3 is enough to explain most of the relevant dynamics. And for the delay period, we obtain these 3 principal components, and the explained variance actually remained high all throughout the delay. And for the delay period, we obtain that these 3 principal components are enough, but also maybe a 4th one, but yeah, trying to project a 4-dimensional space into a 2-dimensional screen is yeah, kind of hard, so we stuck to 3. Okay, so what do we see in this projection? We see that all the trajectories start here and they diverge. The acoustic trajectory goes to a certain subspace here, while the tactile trajectory goes in a completely different way. And the no stimulus trajectory remains close to the starting point. And during the delay, we see that the trajectories remain segregated. And we can relate that to the mutual information that we got before. So this area is still like keeping a decision memory. And yeah, I think that's everything we have to explain here. So questions? Good.
Yeah. Well, yeah, I've seen that it's a key evidence that this area in some way integrates sensory input in some subspaces and then maintain the decision in another subspaces. Or
yeah, that's the idea that we see here. Okay, so yeah, this is just one type of linear projection where we see a 3-dimensional space, but remember that we have a plus 900 dimensional space. So we wanted to employ all the types of projections to get a better look of how these dynamics are evolving. And for that, we employed a generalized linear model. And with this model, we obtained weights, basically 4 types of weights related to the tactile presence, the tactile amplitude, the acoustic presence, or the acoustic amplitude. So for each neuron, we obtain these 4 weights. And yeah, we have 4 vectors of 900 weights. We normalize them and we employ them as projection axis. And we obtain this projection over here for the stimulation period. So similarly, all the trajectories start here, and then the acoustic trajectory diverges over here, while the tactile trajectory diverges over here. And we can see that this divergence is pretty much orthogonal related to the GLM axis. What this means is that we can take a linear combination of the neurons so that we only care for tactile responses or acoustic responses. And that's very cool, but be careful because we don't want to confuse this with clusters. So we are not saying that there are neurons that only respond to tactile or neurons that only respond to acoustic stimuli. And we can see here, it's kind of blurry, but yeah, we can see it here. The neurons response portray mixed selectivity, and there's no clusters of tactile or acoustic neurons. We see that in this space. So basically, each dot over here represents a neuron, and the axes are the tactile presence and the acoustic presence. And we see that if a neuron responds to an acoustic, to an acoustic stimuli, it doesn't give us information at all of how it will respond to the tactile stimulus. And yeah, so basically we can see just that this is just a blob of points here with no correlation. Okay, yeah. So that's just for the stimulation period, this projection over here, but we also employed it for the delay period, and we obtained this. So as we saw with the PCA, the trajectories remain segregated, but in this subspace, we actually see a slight convergence. They're moving towards each other. We can maybe relate that to the decrease in variance which we see during this delay period. And also the orthogonality that we saw first in the stimulation is not that pretty in this projection during this period, I mean, because, for example, here, the acoustic response also moves in the tactile axis. So, yeah. Any questions? Should we continue? Okay, so this was for a GLM projection. the next thing that we wanted to study is the similarity between these responses, because as I have showed you, there's a clear division between a stimulus more a sensory response and the delay more decision maintaining response. So for that, we employed a similarity metric of the population vector. So here what we do is we take the average of the population vector across all conditions, and we take the vector of one time. Let's just start with the plot over here on the right. So we take, for example, this vector. I compare its similarity. For this study, we employ the cosine similarity that tells us the angle of the vector with respect to the vectors calculated in each of the time. So we first took this vector over here at the end of the simulation period.
And again, I think that it's important to clarify that this kind of measure is employed to compare a lot of, when you have a lot of dimension in a vector, it's quite difficult to have a measure that compare how similar these two vectors are behaving. So what the people usually do at the population level is to took the whole vector and to see if these neurons at this moment or this population at one special moment is similar to the population at another moment. So, and looking, there are different measures that you can use to employ distance between two vectors, but at the population community, someone that was working quite well, at least, is the cosine similarity. That is basically the angle between these two population vectors. So this is something it's not so it doesn't depend on the magnitude. It only depends if the combination of neurons are in the same direction. So both two vectors with the same combination but different firing rate at the average are going to have the same angle. So the cosine similarity is going to be 0. Okay. One, sorry, it's going to be extremely similar. Yeah. In the case that two vectors are at 90 degrees, so they are orthogonal, they are completely separated each other, we are at 0 cosine similarity. Okay.
Yeah, that's basically the gist of it. And yeah, of course, we see a similarity of 1 when we take the vector with respect to itself. But here in the sensory case, we see that this similarity decreases in all the other moments of the tasks. So here is low, here during the delay is low, but also after the probe up. If we take, for change, a vector right in the middle of the delay period, we obtain this. So the similarity is high with itself, but it's also high with the rest of the delay. Kind of, more like this. And also we see a great decrease in similarity with the sensory part. Okay, so what we did next is instead of taking just 2 vectors, we take each vector in the time and compare it to every other each vector in the time. So in the end, we get a matrix. That's the one here on the left. And here in this matrix, warm colors represent high similarity.
Which is the dimension of this matrix?
Yeah, it's time by time. Yeah, you got chopped away. So yeah, it's a square matrix, time by time. Warm colors represent high similarity, and cold colors represent low similarity. And yeah, we have to remember that this is the populational rate vector. So the most we can get is a similarity of 0. We cannot get a negative number because the firing rate is always positive. Okay, so what do we see here in this matrix? Here we have marked all the important moments of the tasks, and we see that the sensory response happens here is highly dissimilar to the rest of the task. Over here. During the delay, we actually can see like a block of high similarity. So all throughout the delay, the neuron, seeing the population vector, seems to have kind of the same angle. And this similarity decreases again for the response after the probe up. And yeah, let's remember that this is averaging across all the conditions, but what happens if we divide it across tactile, acoustic, or no stimulus trials? Well, we get these matrices over here. So they're kind of similar, but there's, again, a lot going on here. So first, for this tactile matrix, we see that the response is highly dissimilar during the sensory part with the rest of the task, but not here. And that we can attribute to the probe up. So the probe up, the stimulation that makes the monkey report its decision, is nothing more than a sensory stimulus. So related to the stimulus that the monkey got earlier, it makes sense that both of those states are similar. And this does not happen with the acoustic stimulus.
But your response after the PU is not as high at the beginning, right?
Yeah.
I don't think, I mean, I don't see what you say. Like, I don't see that it responds again because of the stimulus, tactile stimulation.
Okay, I think you're trying to say that it's not like exactly
as
the same
stronger
thing?
as before, yeah.
Yeah, it's not as strong as before, yeah, of course.
And of course, it's like 3 seconds, right? It's like from the beginning, it's, from your stimulation is 3 seconds, right? After stimulation?
Yeah. I don't have the time here. I have it.
Yes.
Here?
Right.
So here, right?
Yeah. So your neuron, if it's the same neuron, if I understand well, the same neuron, right? Yeah, yeah, it's the same population of the same neurons, right? Yeah. So yeah, I don't think it's just because of this tactile stimulation. I mean, you have enough time for release that neuron for the first stimulation. You know what I mean? So in theory, if it's a sensory neuron, I mean, like the modalities, yes, like it's just sensory, or is the most important information that this neuron is carrying on, then you should have the same response.
Okay, yeah. As we see before, well, the BPC doesn't have just like clear sensory neurons, but it has neurons that respond in a kind of sensory fashion. Yeah, they don't need to be the same, I think. Yeah. Yeah.
The same neuron, or they are just changing the behavior. It can be the same neuron, but the neuron can change the behavior, or I don't know, but it's not the same response.
Yeah, I agree.
And same with the acoustic.
With the acoustic?
Right? Like you have, like you have also like this orange color, but it's not as stronger as before, right? Like it's red.
Yeah.
So just a comment.
Yeah. So the responses are not the same, but similar. Yeah. So what we obtain with this metric. And yeah. So and finally, to explain the absent one, during these trials we don't get any stimulus, so the population similarity remains high all throughout the task until the probe up. But we see. We saw with the previous neurons that the firing rate does not remain necessarily constant throughout the trial. They can show ramping. So what this tells us is that these ramping neurons, what they're doing is that they're not changing the angle of the population vector, but rather changing its magnitude only. And yeah, I think that's it.
That's something that is quite unexpected in some way. I have, in the sense that during absent trials, like the network remain quiet. Well, quiet or at least changing in magnitude. Changing in magnitude.
I think maybe that just means that it remains in the same area or direction of the space, so the dynamics are stable during the absent. It's not switching what type of dynamics each neuron is representing.
Yeah, and it makes sense because the neuron, you can have some neurons respond faster, suddenly switch because there's no stimulation, so they can only like remain moving in the same direction as they started. Yeah. Yeah, of how similar is, yeah.
Why you didn't see the opposite? It will be the opposite, so forget it.
Yeah, okay. Okay. So yeah, as we see, most of the dynamics here are time dynamics. So the next thing we wanted to do is actually separate this time or bimodal dynamics from the modal dynamics. For this, we employed the mixed principal component analysis. And what this analysis does is that it basically marginalizes the covariance matrix for modal and amodal responses, and it lets us distinguish between axes that explain modal or amodal dynamics.
Yeah, something that is, I believe, important to remark here is that this approach, this methodological approach, appeared like, I don't know, how many years, 10 years or even 8 years ago. So from this moment, we as community, we were able to separate population responses into focusing on different parameters. For example, looking at decision, looking at stimulus. So it's a kind of PCA, but focus on one parameter. So in some way, they help us a lot to understand how the different signals interact each other, or how the stimulus signal, the decision signals interact. This is something that we needed because, as we were discussing before, the neurons sometimes switch. So it's not so easy to choose axes that in some moments, so we need to make axes that focalize on one parameter especially to try to separate how this is coded across the population. And it's not so easy to do it using only PCA, even if PCA, GLM are really good approach to, in some way, try to understand how the network works, to focus on something. The mixed PCA was like, something that, a revolution in some way for the population analysis or something like that.
Yeah. So what we obtained with this, the mixed PCA was principal components, and similar to PCA, the first ones explain more variance than the latter ones. So here we see that the first three components that explain the most variance are amodal. And this is similar to what has been shown before, that in biological networks, the time dynamics that takes most of the cake when related to the explained variance. But here, the component number 4 and 5 were modal, and followed by amodal components. So we showed, as we did with the PTA, the first 6 components, and yeah, we obtained this. So basically, this the ones on the top are the amodal components, and also this component number 6. And we see that they mostly represent the sensory response here, here, and here. And this component number 2 and 3 actually have the switch. So it starts positive, but then it goes negative. And the six yep.
All components indicate, even if these dynamics are representing the sensory signals, they are all together in the sense that acoustic and tactile are all together. So it's a sensory, a modal signal, something that is saying the network, okay, we are going to start, we are receiving something or something like that. So it doesn't depend, the 1, 2, 3, and 6, it doesn't depend of the modality. So it's like a underlying dynamic associated to a modality or something like that, something like that. The network is solving something associated to the sensory input, or
yeah, exactly. And if we look at the model dynamics, here the component number 4 and 5, we get some actually pretty stuff that the acoustic trials go to one part and the tactile trials go to the other part. And here component number 5, we see kind of this a similar thing, where we also see a switch. And during the delay, they continue to be segregated, but they switch. And what we wanted to do with this two model components was project the activity on this like space of 2 dimensions. And we got this, that is, I think my favorite result so far. So we have these components over here, number 4 and number 5, and we see that the trajectories all start here, and they diverge. The tactiles goes over here, and the acoustic one goes over here. And then during the delay, there's a quick switch over here, and that's represented with a rotation. And the next evolution of the dynamics during the rest of the delay happens in an axis that is orthogonal to the first sensory escapade. So yeah, that merges good with our previous results that the stimulation and the decision maintenance dynamics are dissimilar, and in this case, orthogonal to each other. And we can also see that the trajectories remain kind of like stable in this subspace. So we can maybe postulate the idea that there's an attractor over there, keeping the trajectories segregated.
Yeah, well, I believe it. Oh.
How many neurons do you have, or units do you have there?
Yeah, around 900 or something.
Yeah, but how many are the blue one and how many are the same?
Yeah, they both have tactile and acoustic trials.
They are 900. That.
Something really important what she asked is, even if we have 900 neurons, and even if the neurons are apparently doing something mixed, something heterogeneous, something that we quite not understand, the network in some way go from that direction through the sensory input, and they go to an orthogonal direction to maintain the decision. So the people could ask, why do the brain do this stuff? Well, in some way, it's important for the brain to separate a cognitive process. So when you receive the sensory input, it's important that when you maintain something, it's in a different axis. So why? Because there is like a lot of really recent evidence that we have that the network try to diminish the interference between memory and sensory input. Why that is important? Well, because if we are maintaining something and we need to be able still to represent sensory input, it's something that need to be orthogonal, need to be separated. It's not, so if you are representing something in the same way that you are receiving the information, well, this is going to make some kind of interference, and this is not going to work because so when you are receiving input, the memory is like in some way interfering with the sensory input. So this is a little the idea that in this case it's really great how the network appear or emerge. I don't know.
Yeah. So finally, we asked if this dynamics that we observe in the BPC could be actually kind of universal for solving a bimodal detection task. And in order to answer that, we took the approach of modeling this task.
Your next talk, I think that you can put some axes over. Show this slide and then some axes that are orthogonal above the.
Yeah,
one.
I cannot do that like in the end, but yeah, sure. So yeah, we next employ the model of artificial neural networks, specifically recurrent neural networks. And these models have been shown widely over the last recent years to show very similar responses to what we see in biological networks. And these models are mainly described by these equations over here. So briefly stated, we have a trainable recurrent weight matrix, a trainable input matrix, a bias, and noise that's added at every time step. Yeah, in the case of this bimodal detection task, the main inputs were two units that represented each of the possible modalities. In this case, we labeled them as stimulus 1 or stimulus 2. And for the readout of this network, we have this equation, a trainable output matrix and a trainable bias. And we have 3 nodes, each corresponding to the possible decision or the button that the monkey can press. So what we do is we have over here the time, and we want the network to remain silent in all of the nodes all throughout the task until a go cue is presented. So over here we have what we want the network to produce, and the distinct shade of the colors mean what the network produced after it was trained. So when we present a tactile stimulus over here, the presentation takes place here in time 0. We don't want the network to respond until a go cue, where the network rises the activity of only one of these channels. If we were to have an acoustic or stimulus 1, we want only this channel to rise. And if we don't present anything, we want only this channel to rise. Okay. And if that's clear, we measured the performance of the network. It was able to solve the task. And we obtained this kind of psychometric curves with the probability of saying yes. Here in the x-axis is the amplitude, but it's kind of arbitrary units. And we see that, surprisingly, even though we trained the network in a completely symmetrical way between the modalities, the network responds in an unsymmetrical fashion. Also, when we analyze the trajectories, we see that they are not symmetric. And that's pretty interesting because the BPC also showed a dissymmetry between the tactile and the acoustic responses. So yeah, that's an interesting thing to notice.
Surprising, or really surprising, when I saw for first time this result, is that I don't know why the brain has well, probably because the different inputs or whatever is not so symmetric, the acoustic and tactile responses. But when you train the network, that is, yeah, we have so many evidence that this network, we don't understand why they reproduce a lot of dynamics so similar how the brain does. But this is something even harder for me to believe, that they are able to reproduce the asymmetry between modalities, so how the brain in some way receive one input different or treated input different depending of if it's a tactile or acoustic input. So I don't understand how is that, and I don't know if Bernie has an opinion, or Juan Claudio,
maybe it's just hard to be symmetric, and maybe even if you want to be fair, you always end up preparing one child instead of the other one. So yeah. It's surprising.
with generated random data, right?
Yeah.
This.
Yeah.
So that's why maybe it's impressive that it's that perfectly symmetric. And the brain could be many things, like you said, but here this is just a computer model with symmetric things. I mean, one neuron or the other, when they're the same, they don't respond. No, I have no idea.
Okay, so looking at the trajectories, we employed PCA as well, and similar to the BPC, we get a divergence of the trajectories of the modality 1, modality 2 in the sensory period, and they continue to remain segregated during the delay period, and making actually a rotation. So in these projections over here, we see a very similar thing to what we saw in the BPC. A first sensory escapade, and then a rotation, and then the rest of the dynamic remains kind of orthogonal to that first sensory escapade. So yeah, the BPC and the network dynamics seem to be very similar in this case. And we also look at the similarity matrices. For the ASAP trails, we got kind of the same thing. So, yep.
The first, yeah, there are a lot of confirmation that we have that these networks are so amazing to represent what the brain is doing, and apparently it's quite every time that this is applied to a new task, it's amazing how well this model represents what the brain is doing. So I don't know. This is the first scene that I have seen here in Mexico, but on the other hand, on a lot of people across the world has emulate this different cognitive task employing this network, and it's so so clear how, or so amazing how it's so similar what we see on these models and on the brain. So this is something that usually happens in science, that we have starting to have a model that is able to reproduce, or to be able to understand how the cognitive process emerge. So, and why it doesn't, why these kind of approaches are important? Well, to have a model is always important to manipulate, to make some kind of prediction, and to see, well, in which cases this model is not working well or whatever. So this is how science evolved, to be able to have a synthesized approach.
And there's also the thing with artificial neural networks is that they're not easily interpretable or explainable, in the sense that it works very well for predicting the outcome, but you don't know why it's so good at predicting the outcome.
Yeah, same as the brain.
Yeah, same as the brain, of course, but maybe in these models which are in theory simpler than the brain, maybe we can try to gain some insight by, for instance, in this case, looking at what the weight matrices look like, and maybe trying to find some structure there, and understanding why it works. Maybe we can learn something about the brain by doing this in these artificial networks. But of course, this is an open problem, not just in neuroscience, in all machine learning and all artificial intelligence, that we don't really understand why these things work so well. But I know maybe there's an opportunity to further investigate here the matrices, the weights, the internals of these models.
Yeah, that's kind of like what I've been trying to do, to study the dynamical system that emerges from the equations, from the weights that the trained networks arrive to. And yeah, it's not a trivial thing to do, but it definitely gives you an insight of how the network is working. Yeah, he said like, yeah, Roman's right. That's very impressive. Yeah. Okay.
So I think that this is a point to move afterward. It's like to try to look at the dynamical system and trying to understand how it's solving the task. And well, perhaps we have a theory why the brain is so similar to the network in the future. I don't know. I don't know. But this is something that we need to move forward to that.
I mean, for instance, you showed that in the brain, the neurons are very flexible. They're very heterogeneous. Are the neurons in this model, the inner hidden layer neurons, also this heterogeneous? I don't know.
Yeah, but I don't think it's good to make the comparison between the neurons and the.
I know they're not the same, but something must be similar in the dynamics of what's happening internally, in the sense of how information flows from the input to the output. So do these networks also use this sort of very high heterogeneity to make this work? Or maybe not, maybe these are very like boring behaviors inside the networks and it's not very representative of what happens in the brain. It could be also, but there's maybe something to look into.
Yeah, definitely. Okay, so you're finishing up with these networks. We obtained the similarity matrix. There you go. Yeah, and during the absent trials, we see something similar to the BPC, a very high similarity all throughout the task until the response onset. And during the stimulation trials, we see something that's kind of similar, a high similarity in the stimulation period, followed by a low similarity in the rest of the task. And during the delay, we kind of get the same blob we got with the BPC, but not quite as well. Yep. We have the questions. I appreciate if Roman could send me the paper presenting the simulation using the neural network.
Yeah, well, there are several that you can send, and in the last years, a lot of people, Merraja Zairi lab, Bruno Aberbeck lab, Krishna Shenoy lab, Larry Abbott, I don't know, Xiaoxing Wan, I'm forgetting important people for sure. All these guys, even Gustavo De, Nestor Parga, people that is closer to us, were employing Churlan. We're employing these or applying these networks to solve. Yeah, so Marcos, we can send to you the review that Bernie was citing across the task, across the talk. But apart from that, there are several important papers that they have employed these networks in the last year. So yeah, Bernie, could you send to Marcos, or I'm going to send it to Marcos later.
Yeah. I think that review for Yang has
a lot of
references.
And this something that is quite important is that this Yan and Wan people or group, especially Xiaoxing Wan, that he was working on that. He's in Columbia University at New York. He was able to show that these networks are able to be multitasking, something that machine learning are not so easy to reach, or not a biological representative machine learning. And so they have a Nature Neuroscience paper in, I don't know which year is the paper, but they show that these networks are able to learn 10, 20 tasks different and be able to solve all these tasks in a similar way how the brain solve it. So yeah, I don't know. There are a lot of work going on with that.
Yeah, sure. So to something up, we have where we propose to be the system that the networks take. So first, I want to present this unimodal system proposed by Carnevale and colleagues. And this is the case for the tactile detection task. So here we have the neural trajectories. And what he proposes is that the neural trajectories approach a separatrix in the possible stimulation window. And if the sensory input is strong enough, it kicks the trajectories to the other side of the separatrix, and they end up in the yes, I felt something, attractor. Otherwise, if the sensory input is not strong enough, because, for example, if there wasn't any stimulus, or the stimulus was very low, then the trajectories remain on the same side and end up converging to the no attractor. And yeah, so this is the case for 2 attractors, but for this task, we actually need 3 attractors. So what could be happening is something like this. We have multi-stability. So here we can have distinct basins for each type of response. So maybe the network starts in the stimulus absent basin, and if the tactile stimulation is strong enough, it ends up in the tactile attractor, and if the acoustic stimulation is strong enough, it winds up in the other attractor. And this we can also see it with here with a state transition. So basically, what we saw here is that the network starts in a spontaneous state and then evolves to a state where it has these 3 possible attractors that is in the possible stimulation window, where we actually can feel well, a stimulus, right? So yeah, we can represent that with a switch from only having 1 attractor, the spontaneous state, to having an absent, tactile, and acoustic attractor. But as we saw with the neural data and also the network, the system doesn't look like as simple as this, but it actually has subspaces. So this is the little diagram that I made. We first start here in the sensory subspace. All of the trajectories start here, and the tactile, in the case of a tactile stimulation, the neurons they diverge over here, and in the case of the acoustic stimulation, they diverge over here. And yeah, this response, this divergence is actually orthogonal, as we saw before, but yeah, I ran out of dimensions, so yeah, that we can represent that. And also important to notice that the tactile response, at least in the VPC, is stronger and faster than the acoustic one. And at the end of the stimulation, these trajectories rotate and end up in this tactile attractor, acoustic attractor, and the absent trajectory remains in the absent attractor. So if we take this diagram over here, these three attractors, if we rotate them, I hope that you can imagine, they're like here. So first we have the sensory escapade, and then the convergence to the attractor that keeps the decision memory in the memory subspace. And yeah. So our conclusions with the population dynamics. Yep.
Gustavo Deco diagram. I mean, that surface is just schematic. We don't have a way to, or do we have a way to try to estimate the shape of that surface? Because that will be extremely interesting to see how the landscape is shaped. Could even show us attractors that we haven't even seen yet. I mean, and also explain why the things go such way or such way. But I mean, you cannot do that with your data because you only get a trajectory, right? You get a single trajectory maybe. So probing the shape of that surface is not something, I don't know how to do it, but it would be very interesting to find ways to try to do that.
Well, you can use
maybe with the neural network, with the artificial neural network, we can try to generate a lot of trajectories, trying to put the system in different parts of this surface and see where it goes from there, and then try to build the derivative of all those trajectories to try to reconstruct the surface.
Yeah, of course. That's something that I'll be trying to do as well, but even this simple model of recurrent neural network lives in a high dimension space, plus I think like around 300 dimensions. So it's still hard to like interpret how the
that's something Juan Claudio answered himself. But well, this is something important for the model, is that after you have a model that is quite representative of what is happening, we can try in some way to test it and to learn from it. And And in that case, perturbating or making a lot of, I don't know how to do it because as Bernie was saying, it's so high dimensional, the network. But so if someone has an idea of how to analyze this landscape, energy, stability, or whatever, yeah, I don't know.
Something that has been done before is they use a low rank recurrent neural network. So they actually force it to be in a low dimensional space, and that's like easier to study. And yeah, we can surely try that. And there's also like many other approaches. So yeah, as conclusions, we have that the BPC response is the BPC sensory response is similar to the decision memory. The acoustic and the tactile sensory responses are mainly orthogonal, as we saw with the GLM, and the BPC networks develop 3 attractors corresponding to each possible decision: tactile, absent, and acoustic. And finally, that the mechanisms observed in the BPC could allow us for a general bimodal information integration and bimodal perceptual decision maintenance. And that's it. I want to thank all the partners in my lab. This is an old picture, but I think most of us are there. And yeah, this is my current master's program. Yeah, so thank you. Any further questions? Yes.
Any of you? Okay. Oh, I see that.
Mm-hmm.
Okay, yeah, as you can see, there are a lot of things to do with this network, with this, yeah, as Juan Claudio get emotional now, he's, yeah, he's so happy with these ideas, yeah. Thanks a lot, Bernie, for coming here and to have your, talk about what you have been doing in the last years, and hopefully we keep on working on that. Everything that you have been learning in Germany is so important. Well, this group is so good for modeling and network modeling, so it's nice to have this kind of interaction, at least for us to understand this kind of topic. So thanks a lot, and see you. I don't know if at the end of April that we are going to have another seminar. I don't know if Marcus is, but if we should, but I don't know. We don't have any name on not yet. Okay. Okay. So